So welcome to Chapter 3, the cell and all its functions and structures. If you look at this drawing of a cell, you can tell just by looking at this thing that you're looking at a very complex organization that carries out a whole lot of different functions. And quite honestly, depending on which type of cell you're looking at, you see a whole lot of different things under a microscope. It's a good idea to get a feel for just how a cell fits in terms of relative size. We have probably just come from a unit where you're looking at the biochemistry within a cell, the small molecules that are actually active in terms of production and use within a cell. But those small molecules are way down at the range of about a nanometer. Those molecules then enter into a cell, and that's a whole another chapter in itself. How do they get in and out? Which ones need carriers and which ones don't? Which ones are soluble, which ones don't? It's coming in another chapter. But for our purposes right now, just realize that we are still dealing with something that is, for most cells, not visible to the naked eye. Animal cells have a range roughly of about 10 micrometer. Micrometer is 1 times 10 to the negative 6 meters. And they might range all the way up to about 100 micrometers. So they're small. And yeah, so often you're going to be using a lot of artistic renditions to try to emphasize just what the organelles within that cell actually are doing. So by the end of this very short unit, you should be able to, and these are your objectives. So somewhere in the handouts you've picked up, you should have picked up the actual government objectives for this unit. Again, I must emphasize that any tests you have will constantly be coming back to just these objectives. So they will be stated in different ways in questions, but this is probably the place to start. Notice a lot of it comes down to the verbs involved. You're actually describing, stating, describing, identifying. A lot of these verbs give you a hint as to what they're actually looking for in terms of your level of understanding. So, lots of resources in this unit. Like always, you have far more resources than you actually need. But again, each of you will find that you need more or less, and some of you need to go through this more times than once, and some of you probably don't need to go through half of it. So again, each of you is an individual, and each of you has the opportunity to decide to learn or not to learn. So obviously you have a textbook. It's a good idea to actually read the chapter. You have something called Raycroft notes, which go into, again, far more detail than you do need to know. A summary of Raycroft notes. You have something called the lecture outlines available on the website. You have study guides, you have animations, and again, animations are a great way, just kind of like the Campbell activities that you need to watch to actually see the cell organelles in action, so to speak, is so much better than listening to a teacher talk about it or even reading the text description. You have practice quizzes, and again, those quizzes, most of them come from every single question from old provincial exams. So if you can answer those excuse me, practice quizzes, you know what you need to know. And then obviously there's this guy Bobby himself that would just love to sit down with you and explain any of this that is causing you the slightest bit of confusion. This looks like I might have been a little bit younger, or was maybe playing with Lady Clairol or something, but yeah, don't be afraid to ask for help. So the truth of the matter is, if you look at those objectives, a lot of it is coming down to knowing the organelles within plant and animal cells, and then knowing their functions. So don't forget the functions. It's one thing to recognize them, say, in a picture, but quite another thing to be able to give the functions of those organelles. And of course, at this point, you're asking yourself, the Wailing Wall, Jerusalem, Orthodox Jews, what's the connection? Point is that at the back of the classroom, on the bulletin board, you have these two posters. So I have a little hint for you. If you were to stand back there, pretend that the bulletin board is the wall of Solomon's temple, and you just stand there and rock back and forth, concentrating, pray if you like, and you honestly should have about 90% of what you need. Eventually, all this comes down to, I had to show you my desktop because the desktop really is my belief in education that anything learned is essentially self-taught. I will make resources available to you. I will be personally available to you, but ultimately the decision to learn, to internalize information has to be your own. So again, I encourage you to do that, and I would be delighted to do anything to help you emphasize that. So it's a good idea when you are looking at a complex organization like a cell to look for an analogy. And one of the common analogies of a cell and its organization is to compare it to a city. So you actually have a very ancient, I guess you'd call it a tutorial on the computer called The Cell as a City. Goes through electron micrographs and compares each of those organelles within a cell to various places and functions within a cell. So I'll give you a for instance. Every city has a city hall. And just as the nucleus of a cell controls the functions within the cell is under the control of City Hall. All of the blueprints and plans for development have to be approved by City Hall. DNA within the nucleus carries out a very similar function by controlling the production of proteins within the cell. So let's stick with that cell as a city metaphor for a second and actually look at some of these connections. So the analogy would be lysosomes. In the cell, they contain hydrolytic enzymes. Hydro, by the way, let's show that again. Hydrolytic. Hydro means water. Lysis means split. So these are enzymes which function in hydrolysis. And they break down and recycle used materials. In the analogy of a city, these would essentially be little suicide sacks, demolition experts in charge of reuse and recycling. I think I need to take a drink, eh? You notice my voice is going? Yeah, beaker. Cell membrane. In the cell membrane is selectively permeable. Those are two very important words when you get to the next chapter on the cell membrane. You'll see that the cell membrane controls what enters and leaves the cell. In the city analogy, this would be similar to basically a fence around a city, or in ancient times, gated communities, or even gated communities today. Chloroplasts. Their function is to convert the energy of the sun into carbohydrates, glucose, and in the city, those would be the solar power collectors. Mitochondria carry out cellular respiration, converting glucose into that energy currency called adenosine triphosphate, ATP. In the city, we would say that these are the power generating stations. Cytoskeleton. Immediately when you see that prefix cyto, you know that you're talking about cell. Cyto just means cell. Cytoskeletons make up the framework of the cell, allowing movement of organelles. The scaffolding around construction sites in a city would be very similar. So, be careful here though. If you actually look at a cell and you say take a picture with an electron microscope, this is what's called an electron micrograph. Not always easy to identify the organelle in actual electron micrographs. So you have an artistic rendition down here of the same organelle. You should recognize that as mitochondria. The mitochondria has these shells called cristae, and around the cristae you have a gel-like substance called the matrix. The reason I threw the 3D glasses in here is that if you go through the Cell as a City tutorial, throw on the 3D glasses and you'll actually see these organelles in 3 dimensions, because that's really what they are. We are putting them into 2 dimensions, and it really loses the significance of these being very complex 3-dimensional structures. So when we say cells, you have to realize that we are talking about potentially a whole lot of different things. We're talking about prokaryotic cells, we're talking about animal cells, and we're talking about plant cells. You need to be able to tell the difference and basically know the different functions and structures within each of those. So again, this is not the only resource made available to you. You really should watch the 2 crash course videos made available. They will go into both the eukaryotic and the prokaryotic cells. Obviously, you need to be able to take an animal cell, after recognizing that it's an animal cell, and be able to identify these little factories within the cell. These factories are small organs, so we call them organelles. And as this continues, I will give you the structure and function of each of them. But for now, just recognize some of the key ones. The nucleus, within the nucleus, you have the nucleolus. You have a membrane around the nucleus called the nuclear envelope. You have a membrane around the cell, for that matter, the cell membrane. You have structures called the endoplasmic reticulum, the Golgi, the mitochondria we've just mentioned. When you look at a cell like that, it's always a good idea to then move on to the similar diagram, now without the labels, and see if you can, in your mind, just rehearse what those organelles are that I just mentioned. The ones that we have left here with the labels, they are probably ones that you don't need to emphasize. I've just kept them in there. The ones that I have put in labels for, those are the ones that you probably need to emphasize. So, can you identify this one? How about these guys? Same thing for the plant cell. So a plant cell, immediately you notice this large central vacuole that's usually full of water, and that's the reason you actually water plants. If you don't water your plants, this shrivels up and your poor little plant goes, ah, and it wilts and falls over and gets very sad. So water your plants, sing to them, and then the large central vacuole will get full. And notice when it gets full, it's going to exert a force, and the force goes in this way, this direction, in all directions. But in a plant cell, you've got a cell wall. I don't know if you noticed in the animal cell, there's no cell wall. The cell wall is made of cellulose. You know what cellulose is? We call it dietary fiber. Make sure you eat your fiber, eat your plants. Very good for you. The point is that the cell wall doesn't expand. So the reason a tulip can stand straight up is because of that pressure, that trigger pressure exerted by this large central vacuole. Other organelles that differ from animal cells are, of course, the chloroplasts. They're green. Sometimes you see little starch granules in them because, of course, their function is to actually make food. You take solar energy and carry out through photosynthesis that conversion of carbon dioxide, water, etc. into carbohydrates. So, moving right on, same plant diagram, but now the labels are taken out. So do yourself a favor, stop the video, and just see if you can actually name some of these organelles. Do be careful, however. You get very used to looking at just one diagram. That can be a mistake because every artist gets paid a different amount of money and has a different view on just how a cell organelle should look. So I threw this one in from Campbell just for your own practice sake. You might want to again go through the Campbell activities that are assigned and then actually see if you can label them. So just to summarize the differences between those two cell types we were looking at, the plant and animal cells. In terms of a cell wall, there is no cell wall in an animal cell, whereas the plant cell does. Chloroplasts, animal cells don't have them, present in the plant, carry out photosynthesis. Carbohydrate storage in an animal is in the form of glycogen. Again, it's a polysaccharide. Another polysaccharide only found in plants is starch. And in an animal, the vacuole is not usually present. You have small little vesicles, but of course I've just mentioned the plant cell has a large central vacuole, and the shape, not terribly important that you know this one. So finally, when we move on to the actual organelles here, make sure you have picked up the copy of the quick tour. It's a better idea to actually write down the various functions of these organelles. As you go through it, you can get this information from a whole lot of different places, but the truth of the matter is the test you're writing is written by me, so probably a good idea if you just stick to the actual objectives. So I'm going to give you exactly what you need, so it's not a bad place to start. Even though you can get a lot of this information from the crash course video on prokaryotic cells, I'm just throwing this out here as a difference between the prokaryotic and eukaryotic cells. Pro means before, karyos is kernel, so you can think of this as before a true nucleus. Eukaryotic, eu means true nucleus. So you basically are made up of eukaryotic cells. Plants are made up of eukaryotic cells. Where you're going to encounter prokaryotic cells are in bacteria primarily. So again, when you get a chance, just quickly peruse the differences between them. The big one in this course is knowing that in a eukaryotic cell you're basically going to have all of the organelles, whereas in a prokaryotic cell there's very few of them. Even the DNA itself is not contained within the nucleus, but is found in a little loop called a plasmid, and some of it free floating in the cytoplasm. That's interesting. In all of these cells, I don't think we've actually mentioned the fact that anything other than the organelles is called the cytosol, and the cytosol with the organelles is called the cytoplasm. So make sure you're aware of that. So one more drink, and we're moving on. So cell membrane, also called the plasma membrane, not to be confused with the cell wall. You don't have a cell wall in an animal cell. Again, it's made up of a phospholipid bilayer. Bi just means two. So you've got these polar heads facing water on both sides, and then these lipid tails which face each other. This end is said to be hydrophilic. Both ends were hydrophilic because they're facing water. Hydro, water, philios, Philadelphia, love. They love water. These are hydrophobic, the tails, because they hate water. They are afraid of water, and so they are lipid soluble. And the key here is that the cell membrane is selectively permeable. It decides what can enter a cell and what can't. The nucleus. Nucleus, in our analogy of the cell as a city, is the command center. It's City Hall. This is where basically all actions within the cell are controlled. This is where the boss sits, the mayor. I'd say the boss, in our context of taking this metaphor far beyond where it should be taken, is the DNA itself. So the nucleus is surrounded by a membrane just like a cell. It's called the nuclear envelope. The envelope has pores, and again, those pores control what enters and leaves the nucleus, and things do leave the nucleus. Nucleus contains deoxyribonucleic acid, DNA, which, depending on where a cell is in its cell cycle, you may see under the microscope as this disorganized kind of chromatin stuff, or you may, during cell division, see them as the chromosomes. Those chromosomes contain genes. The genes are the recipes for every single protein that your body uses. The mitochondria. Mitochondria are organelles in which aerobic cellular respiration produces the energy molecule adenosine triphosphate. This, on the right-hand side, is an electron micrograph showing you the real mitochondria. And again, notice the scale here. We're talking about 100 nanometers, 10 to the negative 9 meters. And this, some artist probably got paid $30,000 to paint that. That is the artistic rendition of the mitochondria. What's really important here is making sure you understand the chemical reaction for cellular respiration. You eat carbohydrates, which get converted in digestion into glucose. You breathe oxygen. That carbon dioxide is a product of the respiration of that glucose. It's basically an oxidation reaction, and besides carbon dioxide as an end product, you produce water. I mean, if you exhale on your hand right now, you're going to notice your hand is slightly moist. That's where that water is coming from. And of course, the key ingredient here in cellular respiration is energy in the form of the energy currency molecule, adenosine triphosphate. So, as you might expect, you're going to see mitochondria, and a lot of them, in tissues that undergo significant cellular respiration. And I think muscles come to mind. Muscles are undergoing an incredible amount of respiration in order to produce energy and burn it quickly. The endoplasmic reticulum, there's two forms, SER and RER. SER stands for smooth endoplasmic reticulum, these guys right in here, and then RER stands for rough endoplasmic reticulum. So the endoplasmic reticulum is known as the rough endoplasmic reticulum, which you're welcome to abbreviate as RER, if these little suckers called ribosomes are attached. So these little spherical protein factories called ribosomes, if they're attached to the ER, we call it the rough endoplasmic reticulum. The rough endoplasmic reticulum functions to take proteins synthesized at the ribosomes. So that's key. Proteins are made at the ribosomes. And then, to be of any use in an organism, they have to be transported out of the cell. And to get to the outside of a cell, you actually have to go through a few steps. So from the ribosomes, they're transported to the Golgi apparatus for packaging and export from the cell through this endometrium system, the rough endoplasmic reticulum. The smooth endoplasmic reticulum, SER, as I mentioned before, has no ribosomes attached, so that's how you tell the difference visually. But in terms of function, the SER functions not in terms of protein transport, but in terms of actual lipid synthesis. So again, lipids include steroids, so cells like testes that produce an awful lot of hormones like testosterone are going to have an awful lot of SER. Lipid or fat synthesis. Lysosomes. I think we've already mentioned that in the cell as a city metaphor, these are the demolition experts. Because this is a factory system, this is a cell that is undergoing an awful lot of chemical reactions. There's going to be end products, starting products, things that have to be modified, and in the process there are things that need to be recycled, reused. So they contain hydrolytic enzymes. Hydro means water, lysis means split, so they force these chemicals to undergo hydrolysis and thereby making them available as recycled, and once they're recycled you basically reuse them. So again, look for organs within a body, for instance, that would have a lot of lysosomes. These would be organs like the liver. Can you suggest why you would find a lot of lysosomes in the liver? Okay, I don't expect any of you are heavy drinkers, but the liver has one of these wonderful functions of trying to keep you alive every time you drink poisons, right? So any toxins like alcohol, and yeah, alcohol is a toxin, have to be broken down, and of course it's the lysosomes that have a big role in terms of that breakdown. Chloroplasts. Again, we're talking about plant cells here now. These are the solar power stations of the cell. They take solar energy and they produce glucose in a process known as photosynthesis. You know what photosynthesis is? Basically, it's the reverse reaction to cellular respiration. You just look at cellular respiration where you took oxygen and sugar and produced energy. Now, in photosynthesis, a plant in these chloroplasts can take carbon dioxide from the atmosphere, water from its root system, solar energy, and produce carbohydrates. And then you come along and rip the carrot out of the ground and you eat it. So that's how you get that energy into your body. Among the other pigments in a plant, and in particular in a plant cell, the chloroplasts contain the green pigment chlorophyll. And you know what, in this course you don't really need to get too worried about the actual name of structures within the chloroplast, but these structures again are very integral to the very complex process of photosynthesis. The Golgi apparatus, also known as the Golgi complex. You are not supposed to just say Golgi. It must be Golgi apparatus or Golgi complex. They're composed or made up of maybe at least a half a dozen flattened sacs, which have a fancy name called vacuoles. When I look at this diagram, I actually think of pancakes. So my image in my mind every time I think of Golgi is some rather heavy person just suddenly sitting on a plate full of pancakes, and you get this indentation, and key for us is realizing how it functions. So what they do is they receive these protein-filled vesicles from the ER, and then they process them for transport out of the cell. If you had to put it into 2 or 3 words, I would say the function of the Golgi apparatus is packaging and export. One of the higher level objectives for this unit is knowing how the Golgi is interconnected in function with the nucleus, the ribosomes, and the ER. So far we have just mentioned, of course, the ribosomes make the protein's transported through the ER to the Golgi, and then the Golgi packages and exports them, but we'll come back to this. vacuoles. These are just containers. On your diagram, you'll see them as little circles, usually used to hold and or transport substances within the cell. You can't have chemicals, a protein that is made, simply floating around in solution in the cytosol, because it basically would dilute it, change its composition, etc. So once a chemical is formed, a protein, it usually is packaged in a container, a vacuole. A small vacuole has a funky little name called vesicle. The large central vacuole in plant cells is filled with water and a whole bunch of dissolved minerals, and is a storage site for molecules in solution or suspension. Moving on to the next organelle. And by the way, don't be afraid of that word organelle. I mean, your body is made up of functioning units called organs, right? And the organs have fancy names like your heart and your liver and your lungs. Same thing within a cell. You have functional units called small organs or organelles. One of those organelles is the nucleolus. This is essentially the nucleus within the nucleus. And what it does is actually contribute to or make ribosomes, or at least a major component of them. The next organelle are the chromosomes. You've heard of chromosomes. You've got 23 pairs of them if you're a human, and you are. They are condensed forms of DNA. Basically, under a microscope, you don't normally see loosely organized chromatin or DNA because we're talking at molecular level here, but you may, during cell division, actually see chromosomes. Polyzones, also sometimes called polyribosomes, because you've already done the unit on biological molecules. You're familiar with the prefix poly, means many. So many ribosomes attached together in a chain. They function in synthesizing protein, just like all ribosomes do. But ribosomes attached to the ER make proteins for export from a cell, whereas ribosomes not attached, that exist in this polysome form, they make proteins for use within the cell. Cells, some of them have cilia, tiny little hairs. Some of them have flagella, long whip-like tails. Hair-like projections that are small and numerous in the case of cilia. If you've taken Biology 11 and you've studied Protista, you will probably remember organisms, unicellular organisms like Paramecium that have these little oars. They use them to move around. They are ciliates, whereas organisms like Euglena have a flagellum, long whip-like tails. Both are involved in cell movement, and both are made up of a 9+2 arrangement of microtubules. So you see basically this extension of a flagellum, or a cilia in this case, and notice that it is arranged in a pattern of 9 pairs around the outside and 2 in the middle if you cut it in cross-section. Again, not a major point, a little bit of trivia, but just in case you wondered what the heck 9+2 means, that's what 9+2 means. The cytoskeleton. Cyto means cell. This is the framework, like your skeleton, that basically maintains the shape of a cell. Framework of structures, microfilaments and microtubules that help maintain the shape of a cell and aid in movement. Centrioles, your last one. You're only going to find these arranged in pairs within animal cells during cell division or mitosis. I am going to leave you with a few diagrams just because one of those objectives I mentioned is being able to see how these organelles are interrelated, right, in terms of function. So without me saying a whole lot about these diagrams, you should probably stare at them at some point, probably before the test, and try to string together a few words in terms of how the nucleus, how the ribosomes, the ER, the vesicles, and eventually the cell membrane are interrelated in terms of function. If by the time you reach the end of this unit and you are totally perplexed by what the heck I'm talking about, see me and I will try to elaborate a bit. And the last diagram is a question. What is the significance of this data on cell size? We used to do a lab where you basically blew up balloons. You had a baby balloon, you had a mommy balloon, and you had a daddy balloon, and then you calculated the volume and you calculated the surface area, and then you divided the surface area by the volume, and you got these numbers down here, surface area to volume ratio. That ratio is significant, and it tells you something very important about cells. So again, I'm going to leave that one with you as a little bit of a challenge. Again, if you want to talk to me about it later, that's cool too. For that matter, you can blow up a few balloons and test it for yourself. Obviously, these aren't balloons, these are like sugar cubes, but the same ratio acts there.